Thank you. So yeah, I'm a technician in Ben's group at the moment. I'm going to be talking about what I did for my Masters by Research project, which was investigating a trade-off between two phages that bind different regions of the LPS. So yes, our target bacterial strain is Pseudomonas aeruginosa, which is an opportunistic pathogen and fourth leading cause of hospital-acquired infections, notably affecting cystic fibrosis patients. And up to half of intensive care isolates are now multi-drug resistant, hence the interest in phage therapy. So a major concern for phage therapy is emergent resistance during the course of treatment. And this most commonly emerges via modifications or masking of phage receptors to prevent absorption. But as a result, resistance via receptor modification is associated with fitness trade-offs because by structurally altering a receptor, you're compromising its normal function for the cell. And we can leverage those fitness trade-offs to optimize our phage cocktails. Firstly, functionally diverse cocktails, which target a lot of different receptors, are less prone to resistance because having to modify lots of different receptors comes with a higher fitness cost. So resistant mutants will arise less frequently. And also, targeting specific receptors that are associated with antimicrobial resistance or virulence allows us to steer the population towards a more treatable state, such as the example with targeting the antibiotic efflux porin OPRM, where the phage targeting OPRM, the bacteria has to modify its antibiotic efflux pump in order to resist the phage and so becomes more susceptible to antibiotics. But in order to construct these cocktails that are functionally diverse, we need comprehensive phage libraries that contain lots of phages targeting lots of different known receptors. However, at the moment, phage receptors are rarely routinely characterized upon isolating phages for libraries because there's already lots of time-consuming steps of phage characterization. And of the presently characterized Pseudomonas phages, they tend to target a narrow range of receptors compared to phages of other bacterial species, largely the type 4 pili or the LPS. With a few exceptions being the one phage that targets OPRM and some that target the exopolysaccharide. So in our work, we developed a panel of clean deletion mutants that lack the most common Pseudomonas phage receptors as a resource for efficiently identifying phage receptors and also isolating more functionally diverse phages. So first we screened 50 phages from the Citizen Phage Library, which is the collection we have down in Exeter, against our panel of receptor mutants. And the mutants included the type 4 pili, O antigen, outer core LPS, and OPRM. If a phage was able to infect the parent strain but not a deletion mutant, it indicates that that deleted structure served as the phage receptor. And we found that all 50 of our phages required either type 4 pili, O antigen, or outer core LPS, so the most common ones, and none of them were using OPRM. So our next aim was to diversify the range of receptors that we're targeting for Pseudomonas in the Citizen Phage Library. And to do that, we used a HSDR PIL-A GAL-U deletion mutant as an enrichment host for phage isolation. So through the PIL-A and GAL-U deletions, this mutant lacks the three most common receptors, type 4 pili, outer core LPS, and O antigen. So by enriching for phages on this host, we're favoring the isolation of phages that bind more unique receptors. And also through the HSDR deletion, this host lacks the type 1 defensive restriction endonuclease, which normally protects against phage infection by chewing up injected phage DNA. So this is overall increasing the promiscuity of this host to get better isolation yields and help in isolating these rarer functionally diverse phages. So in my master's, we isolated 12 phages on this host. And of those, 4 were predicted to bind the inner core LPS. And that is because they can infect strains that have truncated LPS where the inner core is exposed, but they can't infect strains that have the outer core LPS or O antigen because that's blocking them from accessing it. And these phages may be clinically valuable, first because Pseudomonas aeruginosa is known to shed its O antigen during chronic infections. So these strains which have their inner core exposed may be there in infections and not able to be targeted by the more common outer core targeting phages. And also by combining phages that target the outer core and inner core, we may be able to create more robust phage cocktails that are less prone to resistance because there's a lot of selection pressure on different regions of the same receptor. So next we investigated the combination of an outer core and inner core targeting phage. And to do this, we generated phage-resistant mutants, and we extracted and analyzed their LPS and sequenced their DNA. So first, 272 is a broad host range Pseudomonas phage from our collection, and it's predicted to bind the outer core LPS because it can infect strains with wild type LPS. It can still infect a strain that only lacks the O antigen, but it cannot infect a strain where the outer core is lacking. And so looking at it on a SDS gel, in the first lane, we have the parent strain, so the wild type, with a large core at the bottom, and that shadow in the middle is the O antigen. And then the next lane is our outer core deletion mutant. And these 3 are the 272 resistant mutants, which you can see resemble the outer core deletion mutant, and they don't have O antigen. So the mechanism of resistance against this phage is to truncate the LPS so that 272 can no longer bind. Through sequencing, we also found that this LPS truncation was associated with along 100 to 200 kilobase paired deletions in the genome, which encompass the LPS synthesis gene GALU. In 2 of the resistant mutants, the deletion also encompasses HMGA, which converts a red compound to a colorless product. So it results in this distinctive red phenotype on the plates. And also in all 3, the MEX-XY antibiotic efflux pump is within that deleted region. And as a result, the resistant mutants are more susceptible to gentamicin, with the MIC decreased from around 3 to around 0.25 in the resistant mutants, indicating also a phage steering role for these outer core targeting phages. Then moving to 1276, this is one of the phages that we isolated on the PIL-A-GALU deletion mutant and predicted to use the inner core LPS. And its resistant mutants on the SDS page restore their LPS in order to resist this phage. So they're putting the outer core and, in 2 of the cases, the O antigen back onto the LPS in order to shield the inner core and block 1276 from binding. So looking at how this then translates to cross-resistance between the 2 phages, the darker blue squares represent sensitivity to the phage, and the lighter blue squares show resistance. So in the first 2 columns, you can see 272 is susceptible to, the wild type is susceptible to 272, but resistant to 1276, while the PIL-A-GALU deletion mutant is susceptible to 1276, but resistant to 272. And our 3 272 resistant mutants have all become resistant to 272, but at a cost of becoming sensitive to 1276. And this is because they're truncating their LPS, so now the inner core is exposed to 1276. And then at the same time, the 1276 resistant mutants, the two which had the completely restored LPS with the O antigen, are becoming resistant to 1276 because their inner core is shielded. But now the residues which 272 binds are restored, so it's become sensitive to that. Taking it a step further, when you take those resistant mutants and expose them to the alternate phage, we see that 272 resistance through the long deleted regions is maintained when you remove the 272 selection pressure and expose it to 1276, which shows that that resistance through those long deletions is maintained in the genome, whereas the resistance to 276 seems more transient because when you remove that selection pressure and instead apply 272, it switches and becomes sensitive to 1276 again. So to summarize all of that, the wild type strain has a complete LPS, is susceptible to 272 and resistant to 1276. And so when you expose it to 272, it truncates the LPS, making it 272 resistant but susceptible to 1276. And this is mediated by long genomic deletions that are maintained in the genome. And then the Pillay-Gao-Yu deletion mutant, which has a truncated LPS, is resistant to 272 but susceptible to 1276. And 1276 exposure produces two resistant phenotypes, one with a completely restored LPS, which is susceptible to 272, and one with an intermediate LPS, which is resistant to both. So as there is a trade-off and resistance to one phage is providing susceptibility to the other, we tried simultaneous treatment with both phages in liquid culture and found that from single phage treatment, so that's wild type with 272 on the left and Pillay-Gao-Yu with 1276 on the right, resistance emerged around 24 hours, but with simultaneous treatment with both, we never saw resistance emerge within the 72-hour window. So in summary, we developed a panel of clean deletion mutants for efficient receptor identification and also for enriching for functionally diverse phages, including inner core targeting phages. We then identified a trade-off in resistance to phages binding the outer core and inner core LPS, and simultaneous treatment with these phages suppresses resistance for at least 3 times longer than individual phage treatments. There's a QR code there for a preprint about the HSDR deletion. And thank you to my supervisors and our lab group. Thank you.